So this is the second video lesson on genes and pattern of inheritance, and today we're going to be looking at the father of sort of genetics and its inheritance, and that is Gregor Mendel and some of the laws he's come up with. Primarily today, the first law. So Gregor Mendel was an Austrian monk who came known as the father of genetics. He formulated laws of heredity upon which modern genetics is based. During his time, it was generally believed that parental traits were somehow blended together at fertilization to provide the traits of the offspring. Although Mendel had no knowledge of genes or chromosomes, his work with garden peas gave rise to the concept of the particulate inheritance. So according to this concept, it is discrete particles within cells which determine the characteristics of an organism. Mendel had discovered and described units of heredity that later became known as genes. So in Gregor Mendel's experiment, he worked with pea plants. Now the reason he chose pea plants were because they could be grown outdoors in large numbers. They were an annual plant, so they produced seeds in one season. Many traits which were heritable occurred in distinct alternate forms, such as tall and dwarf, smooth and wrinkled peas, different types of flowers, and the shape of the flower allowed pollination to be controlled. So important features of his work were that he investigated a large number of plants. He investigated them one at a time, each trait one at a time. He started with true breeding plants, and he carefully controlled the population. Now keep in mind that Mendel's results were published in 1866, so this is quite a long time ago. So pure breeding plants are those which, when self-fertilized, always produce identical offspring, generation after generation. So for example, the seeds from pure breeding plants with white flowers only will produce white flowers. So Mendel's first law was the law of segregation. So here we have his take on this law. So early characteristics of an organism is controlled by a pair of factors. When gametes are formed, the factors segregate or separate. In other words, only one of each pair is present in a gamete. The factors are particles which are non-blending. In other words, their effects cannot be combined. One factor will mask the effect of another factor. Now, these factors are what we now know and now refer to as alleles. So, one trait that Gregor Mendel worked with was the height of the plants. So he crossed to pure breeding. In other words, we now know meant that they were homozygous rather than heterozygous. Two plants, one for each trait. So the gametes produced by the tall plants all contained the dominant gamete, and the dwarf plants all contained the recessive allele in its gamete. And the first generation, all of the plants were tall. We would now describe these as being heterozygous. So in our Punnett square, for the second generation, both male and female gametes were containing one of each one produced. So the male produced one gamete which contained the dominant allele and one gamete which contained the recessive allele. The same was true for the female. And as a result, the following cross takes place. So we have homozygous dominant, we have two homozygous recessive, and one homozygous recessive. Sorry, I'll say that again because I think there's a mistake there. One homozygous dominant, one homozygous recessive and two heterozygous. So this one here and this one here are both heterozygous. This results in a 3-to-1 ratio, which is expected, and we now sort of expect this ratio to pop up a lot. So it is also possible to use a back-cross test to determine the genotypes of the offspring. So both of these tall plants are obviously tall. They contain at least one of the tall factors or tall alleles. Although these are both phenotypically the same, they are genetically different. One is homozygous dominant, the other is heterozygous. In a test cross, the homozygous dominant will only produce one kind of gamete, whereas the heterozygous produces two types, one dominant and one recessive. So we can use a back-cross test to determine the nature or the genotype of the parent plant. To do this, we cross both with a homozygous recessive plant. Now we know its genotypes because it is the recessive version, so it has to be small t in this case. If the parent plant in this case is homozygous dominant, it will contain two homozygous, it will contain two dominant alleles, one in each gamete it produces. Half of the gametes produced by the heterozygous will contain the dominant allele, the other half will contain the recessive allele. When they are crossed with each other, homozygous dominant will only produce tall offspring, while heterozygous will produce 50% tall and 50% dwarf. And that is the first of Mendel's first laws of inheritance. Okay, so that is the law of segregation of factors. Each characteristic of an organism is controlled by a pair of factors. When gametes are formed, the factors separate out, and only one of each factor is present in the gamete. The factors are particles which are non-blending, i.e. their effects are not combined, and one factor, i.e. the dominant one, will mask the effect of the other, i.e. the recessive one. So that's Mendel's first law. We also looked at the back-cross test, which he used to determine the nature of the offspring in the second generation. Okay.